So let's get started. Since we have a long talk and a lot of questions ahead of us, I'm sure, let me know how the audio is. If you're having trouble hearing or anything like that, you can put it in the chat. So welcome. Hello, everyone, to our first of our guest lecture series for our Scholars Club. This is part of our continued effort to educate ourselves on key topics and also collect and document key research and information surrounding budgie care and wellness. Super excited for the first speaker for our guest lecture series is Dr. David Phalen. I know you guys have seen me reference his publications and research before. He has been working in avian medicine for a very, very long time. He's a professor emeritus at the University of Sydney, and also is an adjunct professor at Texas A&M. He spent a very long part of his career studying avian medicine, as well as focusing on health topics for budgies. So definitely going to be really relevant to what we're all interested in. A couple of notes about where to put questions and things. So anytime, feel free to put your questions in the chat. I'll note them down. And then if they're the same question a couple times, I'll collapse them for when we do the Q&A. If when we're going through the talk, there's anything where you're like, I don't really understand, can we go over that again? Go ahead and put that in the chat. We can certainly hop in and explain that a little bit more. This session is being recorded, so we're going to have this recording for afterwards as well. During the lecture portion, we'll all do videos off except for Dr. Phalen, so that the recording shows just his video and his slides. And then when we go to Q&A, we can all hop back in. The structure is going to be 30 minutes lecture. We'll take a quick 5 to 10 minute break. And then just to give everyone a little breather, get some water, we'll come back, do another 30 minutes lecture, and then we'll go to Q&A. Does that kind of make sense? So yeah, that is what I have for today. I'm super excited to get started. I will go video off and hand it to Dr. Phalen to share his slides.
You've got I'm still disabled the screen sharing.
Oh, yes. Let me fix that. That should work now. Yeah. There we go.
All right. Well, first of all, Amy, it's an absolute pleasure to be here, and thank you so much for inviting me. And it's great to see the people that have joined us, and hopefully this recorded lecture will reach others as well. So we're going to start out with my talk called Macrorhabdus ornithogaster. And again, if we can save most of the questions till the end, that would be great. But if you're feeling like you're totally lost, then please don't hesitate to interrupt. All right, so the material we're going to cover today is a history of Macrorhabdus ornithogaster and how it was finally came to be characterized. Where does it live in nature? What species does it infect? How is it spread? What are the signs of infection? What diseases can it cause? How do we make a diagnosis of infection and disease? And what treatment options and non-options are available out there? And is the possibility of actually not treating a bird an option? An infected bird an option, and what's the future look like for long-term solutions to this problem? So one of the things that I'm going to do, here we go, is talk about something that has to do with diseases in general. And that is, diseases do not occur in space by themselves. There is always a number of other factors that contribute to it. And we teach our veterinary students this concept of HEPI, or host, environment, pathogen, interaction, which basically says that you can have a pathogen, you can have a host, and you can have an environment, but unless all three of them are exactly what we need, and we have what I call the perfect storm, where the correct overlaps between the host, the pathogen, and the environment all occur, you will not get disease. So sometimes in order to control disease, we don't have to necessarily get rid of the pathogen, but we can modify the environment or make the host more healthy in general, or modify the host, which we'll talk about how we do that later, and the disease will go away, even though the organism is still present. So that's something that we'll come back to several times during the course of this lecture. So another important factor is this idea of common sense, of which sadly is sometimes lacking in the world. And we can certainly make things more difficult for ourselves if we don't pay attention to some common sense principles that, and this is the case for all diseases. And so we'll talk a little bit about what constitutes common sense as we go through. And here's an example. So what do we know about diseases? We know that if they're going to be transmitted between one bird and another, or one person and another, the more people or birds that we have together, the more likelihood that there is going to be disease transmission. What do we know about diseases? That the poorer the sanitation, then the more likely the disease that can be transmitted from one animal to another. And in Macrorhabdus ornithogaster, we know that it's in the droppings, and that if a bird consumes something that's contaminated with the droppings, then it can become infected. And so if we have a bird that poops and we've got lots of birds in a cage, then there's greater chance of disease transmission. It might be that there's a greater chance that these birds are going to be stressed, which might make them more likely to be infected. If they're in a cage where their perch is right over their water bowl or right over their food sources, then the organism can get into the food and water and be more likely to be transmitted to other birds. So just some basic concept of hygiene and reducing crowding and reducing stress would be things that might help to reduce the chances that Macrorhabdus would be a problem for people and their birds. And then lastly, what I want to indicate is that I can get up and talk about all different kinds of solutions to all different kinds of problems, but unless the solutions are practical, unless there's something that you can do yourself at home, or then there's no point in me even talking about them. And so we have to try to focus on what's practical and what's useful and what can happen. And what might be practical for a person who has an individual pet bird might not be practical on a scale for breeders that are breeding birds for show. An individual pet bird in your house, you might not take it anywhere, and it might not be exposed to other birds, but breeders take their birds to shows. And so they're taking them out of the house and potentially exposing them to other birds all the time. And so we might need to take into consideration the individual and their circumstances when we think about what options there are for controlling this disease and treating birds that have it. So there's a long and sometimes complicated history associated with Macrorhabdus or megabacteria or avian gastric yeast, all the same thing. And it goes back into the 1980s, the early 1980s, when people were first starting to see this in canaries in Europe, and then also in several different species in the United States. And then it was recognized in Australia by a friend of mine, Lucio Filipich, in the late 1980s. And he was a budgerigar breeder, and he was quite interested in how this may have moved around. And he found out that, of course, that budgerigars had been sent around the world, and then we'd selected for the show type budgerigar and all the characteristics associated with that, which they didn't have in Australia. They had the native form, and they re-imported them into Australia, oftentimes illegally. And he thinks that that re-importation may have been the way that Macrorhabdus got back into Australia. But what we know, no matter how it spread around the world, is that it is everywhere, that people have budgerigars, canaries, and many different species of finches. And we're having people that are studying this in the Middle East and Iran. There are some scientists that have been doing work on this, and they're seeing it in budgerigars in the Middle East, and canaries and finches as well. We have scientists, some very highly qualified scientists in Japan that are studying this, as well as in Australia and in the United States. So what is this organism, and what does it look like under the microscope? Well, first of all, it's a yeast, and we'll tell you how we got to that point in a minute. And most people that have looked under the microscope for yeast, if you're a medical a person that works on humans or dogs and cats or whatever, thinks of a round structure, and that they're typically budding when they divide, and that there's a round structure that has a little bulb on the end that comes off of it, and it gets bigger until there's two of them the same side and they separate. But Macrorhabdus is not round. It's this long, elongated structure, which the Dutch, and I can't pronounce how they named it, who are the first ones that really studied this to any degree, called them, that in Dutch, they called them yeast sticks, or essentially, which was a term that was used to describe the French breads, the long, crusty French breads. And you can see why that might be the case. They're long organisms that are very thin, and they have a sort of a rounded edge. And this is what they look like under the microscope when they're just mixed with a little bit of water. A scraping or poo is mixed with a little bit of water or saline, and put on a slide, and then covered with a cover slip. And then they're put on the microscope, and there's a condenser on microscopes that brings the light down a little bit. There's a stage condenser, and with the stage condenser down, then the Macrorhabdus show up very nicely just in the wet preps. And we can do special stains on them. Most people don't typically do these. This is called a periodic acid stain, periodic acid shift stain, which is what we usually use to stain fungi. And you can see it stains nice and red, which would be consistent with Macrorhabdus being a fungus. This is a special stain that we did that highlights nuclei or DNA inside of a cell. And we can see that the Macrorhabdus are actually multiple organisms in a row. They're just still hooked together. They come apart eventually. And that there's multiple nuclei that are demonstrated by the arrow in that slide. And then they can be stained with the typical stains that we use to look at fecal feces in sick or healthy birds. And one of those is called the Diffquick. And the slide down here, there we go. This is a Diffquick of these slides of the Macrorhabdus. And they stain variably with this. This is a really good stain. This is a textbook stain, which means we can never reproduce it again anywhere else. But textbook is what they put in the book, but it was hard to duplicate in your own hands. Anyway, if the Diff-Quick works well, this is what they look like. But when the Diff-Quick doesn't work all that well, sometimes they just have sort of granular staining throughout. And it's the same thing with the Gram stain. They'll have this fine granular staining of the inside of the cell and not the outside of the cell like we see with bacteria. Gram stains were first developed many years ago by a man, surprisingly, whose name was Gram. And he made them so that he could differentiate between gram-positive organisms, which stain purple, and gram-negative organisms, which stain pink. And most yeasts will be gram-positive, and Macrorhabdus is, in fact, gram-positive, and we can see it right here. Okay, so Macrorhabdus is a very big organism compared to a true bacterium, and it's been called a megabacterium, and we'll see that that was not the appropriate thing for it to be called, and we shouldn't use that anymore. But if it was a bacterium, you could see it would be quite large, because these are what a normal size gram positive rod looks like in the droppings of a bird, and they're much smaller, of course, than megabacterium, which is not a bacteria at all. But when it was thought to be a bacteria, you could see why they called it mega. Okay, so we now know that it's a yeast, and the name of it is Macrorhabdus ornithogaster. And a lot of budgie owners and aviculturists have a problem with that. They like to call it mega because that was easy to do. And I say, well, don't worry about it. You know, we can just call it macro, and that's just as easy, or Avian Gastric Yeast, AGY, if you want to. But you don't have to keep trying to remember Macrorhabdus ornithogaster. You can just remember macro, and that would be fine. So we don't use megabacteria anymore, and that can be quite misleading. But why did we think it was, how did it get that name? Well, it happens that Dr. Van Herk, a Dutch scientist who was studying this in canaries, was quite convinced for a number of different reasons that it was a bacterium, and he's the one that first coined the term megabacterium. Now, his graduate student was a gentleman named Dr. Dornstein, Jerry Dornstein, who's a lovely guy. And he had some additional data at the time that he didn't really think it was a bacterium, because he'd looked at these organisms with the electron microscope, which means that he could magnify it 20,000 times and look at the internal structure of the cells. And he thought it was more consistent with it being a yeast, but he had to go with what his boss thought. So it was originally published with the term megabacterium. And then in the late 1980s, Dr. Graham, who was my mentor and a microbiologist in our department, were able to grow a bacterium from the stomach of budgerigars. Now, the bacteria that they grew did not keep the same shape and even the same staining characteristics in culture. And it turned out that they grew a real bacterium, but they did not grow Macrorhabdus. And they had made some mistakes in their observations. And so they published this work on this bacterium, which they thought was Macrorhabdus, but in the end, it wasn't. And we went back and tested their cultures, and in fact, they grew a bacterium and not Macrorhabdus. So all the work that they did turned out to be erroneous. And so if anybody says that it is sensitive to antibiotics, and refer back to this paper, that is no longer true. So our final proof that this organism was in fact a yeast was done in my lab by my research associate, Betty Tomaszewski, and some others that we worked with, and we were able to purify the organism. We weren't able to grow it initially, but we were able to purify it and then extract the DNA from the organism and sequence part of that, and then compare that to the DNA of other yeasts, and we were able to show that in fact it was a yeast. And so far, there's only been one species of this organism found. There's some different strains, but there's only one species that we found so far, and they're very similar all across the world wherever they're found. And then, of course, the question is, why am I going on and on and on about this? Who cares if it's a bacteria or if it's a yeast? And it's really important because we treat bacteria with certain types of medication, and we treat yeasts with completely different kinds of medication. So if we thought it was a bacteria, then we would use the typical antibiotics we use for other bacteria. But because it's a yeast, we need to use antifungal medications. Okay, so one of the next major steps in our understanding about this organism was done by a lovely Japanese lady who came and worked in my laboratory. She had a very short period of time to be in my laboratory, and so she was keen to get started. And in the first week we were there, she had approximately 30 or 40 or so different experiments to try to grow this organism, culture conditions that she wanted to try to grow this organism. And of those, all of those different ones, we were successful. And the first ones that were successful at growing Macrorhabdus, and only 2 of those approximately 40 different culture types that we did. And she had, of course, had major jet lag coming into the lab, and was but refused to take some time to get rested. And we went ahead and got this set up and growing. And one night she didn't want to go home. I was offered to take her home. She wanted to work a little later. And I came back into the lab, and there she was, her head down on the bench, completely asleep and snoring away. But within that week, we were able to grow Macrorhabdus for the first time. And it was very exciting because that meant we had a new way to start to study it. And we could grow it and test it in the lab, and we didn't have to be doing our tests directly on birds that were infected. And what we found out about it was, and not too surprising because it lives in the stomach, is that it liked to grow at a low pH, and that it probably in the stomach feeds on some of the juices that are secreted into the stomach by the host. And so it would only grow if we put serum from an animal, bird, a chicken, or even a calf in the growth media. And then, like all yeast, it liked sugar as an energy source. And then, because it's particularly a little guy, it only grows at low oxygen conditions. That only grows with supplemental carbon dioxide added to the growth and to the environment that it's growing in. And it grows best at the temperature of a bird's body temperature, not surprising, of 42 degrees C. So once you get all those things put together, then it grows quite nicely. And here's an example of just one of the graphs that Hannah produced. And you can see that at room temperature, it doesn't grow at all. At human body temperature, it grows fairly slowly, but at the avian body temperature, it grows very, very quickly. Okay, so where does this yeast, where does Macrorhabdus ornithogaster live? And before I go any farther, and you might wonder why that horrible name. Well, Macrorhabdus means big rod, and ornithogaster means bird's stomach. So Macrorhabdus ornithogaster is a large rod from the bird's stomach, and that makes it easier to understand why we named it that. and also makes a lot more sense about why it has that name. Well, a bird's stomach is a fairly complex structure, more complex than our own, and that it has two major parts to it. It has the proventriculus, which we see right here, and that is what we call the glandular stomach. That is the part of the stomach that produces the acid, which helps to break down the food that they eat, and the enzyme called pepsinogen, the enzyme precursor pepsinogen, that when it's in contact with acid is converted to pepsin, and that also helps to break down the food that they've ingested. And so this thick part of the stomach is filled with glands that all they do is produce acid and pepsinogen and secrete it into the lining of the stomach. And as the food passes through, it's all covered with those acid and the enzyme. And then they pass through to the second part of the bird's stomach, which is called the ventriculus. And that is the grinding stomach, or the ventriculus. And some people like to have call that the gizzard, and they take chicken gizzards and fry them up and eat them. That's a common delicacy in the South of the United States. But it's the grinding stomach. It's very, very muscular, and food moves in there, and then it gets ground up, and when it gets small enough particles, then it moves from the grinding stomach into the intestine. And Macrorhabdus, being the fussy little bugger that it is, will only grow in a healthy bird in one part of that stomach, and that's called the isthmus, which is the junction, the narrowing junction between the glandular stomach here and the grinding stomach right here. So the Macrorhabdus is only found right here in a healthy bird. In a sick bird, it may grow further up into the proventriculus and down into the lining, or the coiling lining of the gizzard. But in a normal healthy bird, and in most birds, even sick ones, it doesn't get much past this point right in here. Okay, so what kinds of birds can be affected with Macrorhabdus? Well, we're still learning, and there's lots and lots of new birds that are being found with Macrorhabdus infections all the time. And some of them are in our avicultural collections, and some of them are in the wild. And we know that the birds that are most severely, significantly impacted by it in our avicultural collections are budgerigars, lovebirds, lesser extent cockatiels, and then finches, zebra finches, and all Australian species are susceptible. Gouldian finches, canaries, and in wild birds we have in the United States, the only place it's been found in wild birds so far is in white ibis, of all things, and in Europe, we found it in European finches, like the chaff finch and the European goldfinch and the green finch. And in Australia, we have it in all different kinds of wild parrots over there, and the introduced green finch and the European goldfinch. We don't know whether if Macrorhabdus is present in wild budgerigars in Australia or not. No one's done that study yet, and that would be a great one to find out. We see it in red-capped parrots in the wild that come into care regularly, galahs, other species of cockatoos, king parrots, and ringneck parrots. So red-capped parrot right here. Introduced native finches, and then in Europe we see it in the European goldfinches, green finches, and the bullfinch and the chaff finch in the wild. In the United States, those I've already mentioned, the white ibis which was found by Dr. Hernandez at the University of Georgia. And that's the only wild bird that we see it in so far, at least. And in Australia, people have a lot of aviaries, and so when they talk about an aviary, they're usually talking about a set of cages, or one cage at least, in their backyard where they keep their birds, and most of their birds are kept out of doors. And of course, the wild birds will come down and walk on top of those cages and poop inside those cages, and sometimes walk underneath the cages where the captive birds are being kept, and they can get exposed to Macrorhabdus that way. And the captive birds can be exposed that way as well. And so there's probably a lot of back and forth that's going on in Australia between aviary birds that are kept out of doors and wild birds. And we're increasingly seeing now in Europe and the United States, and particularly in Australia, where keeping backyard chickens is becoming more and more of a popular thing, that we're seeing infections in chickens, and it can make chickens ill. And so they can come into the clinic with signs very similar to what we see in budgerigars, and we can we treat them in the same sort of way. Does everybody know what where budgerigar comes from? Have you talked about that before?
Not extensively.
So it's an Aboriginal word, and the English have always felt like that it meant pretty bird. But if you talk to many Aboriginal people, they will tell you that it means tastes good. So Aboriginal people looked at budgerigars differently than the English people that tamed them for pets. They looked at them as an important source of protein and food.
Fascinating.
Okay. So one of the things that you might hear is that Macrorhabdus can infect mammals. And we really think that that's not the case with an extreme circumstance, which I'll speak to in a minute. And the reason that we think about that is because there was a report in one dog and in one cat in a British journal many years ago, and they weren't able to culture the organism and they just looked at it under the microscope, and they actually didn't even, I don't think they published what it looked like under the microscope. They may have, but I wasn't convinced by reading that. And subsequently, I'm quite convinced that what they were seeing was a type of bacterium or possibly another fungal infection, but it definitely was not Macrorhabdus, because we've done infection trials in rats, and we can't get it to grow inside of a rat. And if it won't grow in a rat, it probably won't grow in a dog or a cat. But there's a special type of monkeys. They're the colobus monkeys, of which langurs are two species, or one. There's species of langurs, which are types of colobus monkeys, and they just eat leaves, and leaves are very hard to digest. So their stomach is more like a rumen in a cow, which has got multiple different chambers, than it is like a simple stomach, like in most monkeys and like what we have. It has only one single chamber. And apparently it has just the right environment in one of those chambers of its stomach where this Macrorhabdus-like organisms can grow. And that organism has been sequenced. It looks much like Macrorhabdus, not exactly the same, but it's been sequenced. And it's clearly very closely related to the Macrorhabdus we see in birds. But that's the only situations where we know of where Macrorhabdus can grow in a mammal. And then the other thing about that is we've already spoken about how it can only grow in a certain part of the stomach. There have been some reports of it growing outside of the stomach, but I have looked at those reports very carefully, and I think that it's quite possible that the organism they were seeing was not Macrorhabdus, and that we have no evidence that Macrorhabdus can grow outside of the stomach of any animal. Okay, so how does one know if a budgerigar has a Macrorhabdus infection and is sick? And so let's talk about what the signs of the disease are. Well, typically we take a history first, and these birds are typically in the age range of 2 to 3 years old when they first start to show signs. Now we know that budgerigars, based on some studies that I've done that I haven't published, but we know that budgerigars are probably infected when they get regurgitant fed from their mom and or dad when they're very young. And if we were to look at their stomachs at the age of about 10 days in a flock where there's lots of the Macrorhabdus about, that the vast majority of the chicks will be infected at about 10 to 12 days old. But they're not sick, and they don't have evidence of disease in their stomach. They just have evidence of infection. And then, oddly, by the time they get to the point where they're ready to wean, most of those birds will no longer be sick. But that some offspring from some parents will or won't be sick, won't be infected anymore. But some offspring from some parents will be still infected at the time of weaning. And so this is where we come back to this idea about HEPI. And this is where we talk about the host. And it's clear that certain individual budgerigars are much more likely to be infected and probably to come down with disease than other ones that sort of just shrug it off. Okay, so when we have a sick bird that's infected and ill, they appear to want to eat, and they will sit on the edge of a food bowl, and they'll bend down and they'll pick up a seed and or pellet, and they'll grind it, and you'll look down to the bottom of that bowl and there'll be lots of ground up food, but they're not actually swallowing very much of what they're eating or what they're picking up and grinding. And as a result, they're losing weight. They get very, very thin. They typically have nausea and vomiting. And I don't know how many of you have ever seen a vomiting budgerigar, but they pop their heads up and down, up and down, up and down. And then some of the regurgitated material comes out. And as their head goes down, the saliva goes up and lands plop on top of their head. So a vomiting or a regurgitating budgerigar will have slime stuck on top of his head, and that's saliva. And then these guys don't feel good because they've got ulcers in their stomach, and they will sit fluffed up on the perch. They'll seek out heat. If you've got a heat lamp around the edges of the cage, they'll go and sit under the heat lamp. And it's clear that they just are not happy campers. So when we looked at the birds that come into the hospital, owners may not notice that they've been regurgitating, but we can see that when we take them out of the cage, we can find that they're typically dehydrated, that their pectoral muscle mass or the muscles that they use for flying have shrunken up. Oftentimes they have soiling of their tail feathers because they will have diarrhea. And if we look in the cage, it's very common that we'll see undigested seeds in their droppings. Now, lots of things can cause these kinds of signs. And so if you take your budgerigar to the veterinarian, they may have a high index of suspicion that, in fact, your bird does have a Macrorhabdus infection and disease, but they probably will do some other tests to make sure that they don't miss something that could cause the same sort of signs, or that you might have more than one thing going on in that budgerigar. And so quite often what they'll do is they'll take an aspirate of the crop. So they'll put a metal tube feeder or a rubber tube down into the crop. They'll flush a little fluid out. They'll suction that out, and they'll put that into under the microscope and look at that. And they'll see trichomonids if they're there. And they're single cell organisms that are very active and moving, and they'll be swarming all over the place. And then we know that that's a part of the problem. It may not be the only part of the problem, but it's a significant part of the problem that we need to treat. Now, they can also have worms of their intestine. And so your veterinarian may take a little bit of the droppings, and in addition to looking at it as a wet prep where he mashes it up, he or she mashes it up and puts it with some water and looks at it under the microscope. They might also do a fecal flotation where they take several droppings and mix it with a solution that lets the eggs, parasite eggs, come up to the top, and then they'll look at that under the microscope, and look for worm eggs in there. And then, because Macrorhabdus isn't the only yeast that can infect a bird's digestive tract, when they do a gram stain, either of a crop flush or a gram stain of a fecal smear, they may be looking for a yeast called Candida albicans that can cause these similar sorts of signs. And in Australia, and I don't know why it's not so common around the rest of the world, but Australian budgerigars and cockatiels and rainbow lorikeets love fibers, and they'll eat fibers off of a towel or anything that they can get, cage cover, and they'll eat it to the point where they get an impaction. And cockatiels and budgerigars get impactions of the crop, whereas our rainbow lorikeets over there will get impactions of the stomach. But when your veterinarian is doing a palpation on they'll first of all ask you if your budgerigar has any opportunity to get any fibers of any sort. And then they'll feel the crop. And if they feel a nice firm mass inside that crop, that'll make them think that maybe we've got an impaction of the crop, and that's what's causing the problem. And if we take a look at the wet mount of a budgerigar that's has an impaction of the crop, we can actually see the fibers in his droppings, and that can help us to make a diagnosis. And then there's other kinds of diseases, and cancers of the stomach and the organs can also result in signs that are very similar to a Macrorhabdus infection. Okay, so the physical exam or the avian consult is divided into multiple parts. So this is what we teach our students, and they take a history, and then they examine the bird in the cage. And one of the reasons that we examine the bird in the cage before we grab it up with a towel and look at it outside of the cage is that some birds can be very, very sick. And even to the point that if you were to catch them, we might push them over the edge and they could die. So we need to make sure that the bird that we're going to examine can tolerate that. And if it can't, then we might give it oxygen or provide it with supplemental heat and give it a little bit of time to get used to its environment before we do a physical exam. Or we might actually just take out some of that poo that's in there and look at that first. And if we see lots of Macrorhabdus or yeasts or other things, we might go ahead and treat it and not even do a physical exam. We might just treat it for the things that we found. Give it a chance to get a little bit better before we do a full physical exam. And then we're going to probably do some specific tests for Macrorhabdus, which we'll talk about in the next slide. And many of these tests that we're going to do also are good because they can pick up other infectious diseases. We've already spoken about the crop wash that we would do in these birds looking for trichomonas. And in special birds that we haven't found anything else and the owner really loves and wants to pursue diagnostics, and we can't find anything that would cause the bird to be showing these signs, we might take radiographs, or people call them x-rays, and we might even collect blood for white blood cell count or biochemistry profile to look to see if the liver and the kidney and different organs are healthy, or if there's any evidence that we're dealing with an infectious disease and what kind of infectious disease that might be. Okay, so one of the great things about Macrorhabdus, not that there's anything great about it, but I'm one of those people that likes immediate gratification. And when I'm in the consult room, I like to figure out what's going on straight away. And so I'm not one of these people that likes to take a test and send it off and have to wait for the results. And so often when we have a sick bird that comes in with Macrorhabdus, we can take a little bit of their feces and we can mix it with some water and put it under the microscope. And lo and behold, there are the organisms. And I go cha-ching, and I go, we've got what we were looking for, and now we know what we need to treat, and get very excited about that. And so typically, most veterinarians will do what we call a fecal wet preparation, where we just take a bit of droppings and mix it with some saline, put a drop of that on the slide and look at that. But there's an even better way to do it, and it's just as easy. And we take a little tube and we put some feces in there and some saline in there, and we mix them together. And then we take a drop of the surface of that fluid and put that under the microscope. And Macrorhabdus, this yeast, tends to float a little bit and doesn't sink down as fast as the rest of the junk in the poo. And so when we collect that drop off the top, we not only concentrate it, but we get it separated from the other things in the feces that might be confusing to look at. And we can put that on the slide and look at that under the microscope. And so I've got 3 stars behind that. That's the best way to find it in the droppings. Wet prep is the second best way. And then the fecal gram stain, or another type of cytological stain, is not as good, because these Macrorhabdus don't like to stick onto a slide and can wash away when you're doing your gram stain. And so there has to be a lot of them for you to see them on the gram stain or the diff quick. But we often do find them that way, but that's not the way that's the most sensitive. And then there is another test that you can do. If your veterinarian is absolutely convinced that this looks like Macrorhabdus, but they can't find them in the droppings, and that's okay, because not all birds, even when they're infected, are shedding Macrorhabdus. And sometimes you have to test them over and over and over again before you can find it. But there is another test that we can do where we can send in a dropping to a lab that extracts the DNA from that dropping. And then they do a test called the polymerase chain reaction, which amplifies the DNA that you're looking for to a level that they can detect it. And that's a very, very sensitive test. And you don't have to see the organisms in the droppings for the PCR assay to be positive. And so that was sort of our last resort. But if because it's a little bit expensive, and we can't get our immediate results, we have to wait a little while to get the results back. But that's an option that we have available to us. Okay, so we do our wet preparation, or our mixture, and then take a little drop off the top of that mixture and put it on a slide. And you can see all sorts of junk in here, but you can also see all these Macrorhabdus organisms in here. And there's lots and lots and lots of them in this one. And that means this bird is shedding lots and lots in his droppings. So if we just see one every now and again, then the bird's infected, but it may not have disease. It just means it's infected. And I'm fond of saying that if you look hard enough in any dropping, and look hard enough and long enough, you'll find something that looks like a Macrorhabdus, whether it's there or not. And that may sound kind of funny, but there are little bits and pieces of things. Like, for instance, in this slide here, we've got these long, very slender rods, which are probably, where's my pointer? There it is, which are probably not Macrorhabdus, these very, very slender bits. And if you look at them really closely, usually they're broken off at the end as compared to what the Macrorhabdus, these guys are. So I never make a diagnosis just on seeing one organism in a sample that I'm looking at. I usually like to be able to see at least 2 or 3 or 4 or 5 organisms in there before I'll be convinced that that's what I'm seeing. So if we have a lot of organisms like we do in this smear, then that means we've got a significant infection. We've got lots of organisms in the stomach, and we probably have a bird that may not be showing signs, but there's things that are going on in that stomach that make us worried. And we would treat a bird that had lots of organisms in its droppings like this under most instances, unless it's too stressful for that bird, and then we might rethink that. But if we have a bird that's vomiting and we see lots and lots of organisms like this, then we think that this is most likely the cause of their sickness and that it's causing disease in their stomach. So if that bird were to die, or if you were to bring me a dead bird, then I would do a post-mortem on it. And what I would be looking at, again, is the proventriculus and the ventriculus. Now, I cheated a little bit before when I showed you that, and showed you the proventriculus and ventriculus of a chicken, which of course is going to be much bigger. And this is the proventriculus and ventriculus of a healthy budgerigar right here. This is the proventriculus, and this is the ventriculus here, the muscular stomach that grinds up the food. And this is the proventriculus and the ventriculus of a budgerigar that has Macrorhabdus infection. And you can see right now that you can see inside the stomach without even opening it up. You can see the seeds. And here's another example of one where the wall of that proventriculus has gotten so thin that it's almost transparent. Now, in others, it'll get thick. And they can either be very, very thin, or they can be very, very thick. And when we open them up, usually when they're thick, there's lots and lots and lots and lots of mucus inside of them. And then there may be little black spots in the lining of the stomach, which are actually ulcers where the blood is leaking out and being digested, which makes it black. And then we know that we've got disease that's consistent with a Macrorhabdus infection. And then what we do, and this is a bird with Macrorhabdus. You can see these little black spots in the proventriculus, and those are ulcers, and he's bleeding from those ulcers, and there's some discoloration of the lining of the ventriculus. And if I were to take then a scalpel blade and to scrape right at that junction between the proventriculus and the ventriculus, right in this area right here, and then look at that under the microscope mixed with a little bit of water, then there would be just masses and masses and masses of Macrorhabdus there, and we could make a diagnosis straight away. So this is the same example, or perhaps another example of the Macrorhabdus that we would see under the microscope of a scraping of the stomach. And then we can also, if we want to, we can send in the tissues to be looked at your friendly neighborhood bird pathologist, who will make sections of the stomach, stain them with special stains, and then we can see the organisms. And these are the Macrorhabdus here, and they line up in what we call a log jam sort of appearance, and they get tucked in there between the glands of the stomach. And this is the stomach of that bird right there. The stomach gland, those are the glands that secrete mucus and digestive enzymes, and the Macrorhabdus are growing in between those glands. Okay, So it might be that your veterinarian if you bring in a dead bird, diagnoses it with Macrorhabdus right then and there, but says, I want to submit some other tissues or want to do some other tests because lots of birds can have Macrorhabdus, but they can also have other infectious diseases as well. And I want to rule out the possibility that other diseases might be present in that bird that died. And they might send in tissues, look for the beak and feather disease virus, might do tests for polyomavirus. They might do scrapings of the intestine to look for an intestinal parasite called Giardia. They might look to open up the intestine and look inside and look for roundworms, nematodes, or they might look to see if there's any infection of the air sacs, which could indicate an infection with psittacosis. And so all of these things might occur in the budgerigar and be unrelated to the Macrorhabdus infection. Although my former PhD student, Hamish Barron, who is a budgerigar breeder, and I would strongly encourage you to check out anything that he's written or published, and you might get him to give a presentation someday in this forum. And he's a great presenter, so and just loves budgerigars. So great guy to talk to.
Could
And
you
he
spell that? Spell his name?
Yes, it's Hamish, Hamish Barron, Baron.
Got it.
And he's one of the co-owners of Birds and Exotic Pets in Melbourne. Or I think it's called Birds and Exotic Pets Vets in Melbourne. He would have a lot of good stuff on their website. He's of the opinion that some of these things might predispose to budgerigars having problems with Macrorhabdus. So, for instance, if we can control some of these other diseases, and we go back to the host environment pathogen interaction concept, if we can control some of these other diseases, we might be able to eliminate Macrorhabdus, or we might be able to reduce its significance and its impact. I'll just comment that what I've just said is that that there are a lot of diseases that are present in budgerigars in captivity, and so Macrorhabdus may be just one of more than one disease that you could be dealing with in an individual bird or flock of birds. And so if a bird dies, then your veterinarian, even if they did find Macrorhabdus while doing the post-mortem, might want to send off tissues or do other tests to make sure that they're not missing something. Okay, next slide. Here we go. What did I do? Got my... Don't want to annotate. There we go. Okay, so this has come up over the years, and I've had some very keen aviculturalists and individual pet owners that have wanted to check their own birds' droppings out at home, and they either have a microscope or buy one. And what is my opinion about that? And my opinion about that has always been that if you have, I'm sorry, I'm going to have to take another drink. Someone that's keen and enthusiastic, the last thing you want to do is to sort of shut that down. So I would say, absolutely, if that's what you want to do. And then, of course, the next thing they do is, well, would you train me how to do that? And I have done that over the years for several people. And I found that some people, after looking under the microscope for a while and practicing, can get very good at that and can find Macrorhabdus and other organisms in the droppings with a microscope in their own home. Whereas others are not quite as good at it. And keep thinking that they're seeing Macrorhabdus and then bring their droppings in and we look at it with my double-headed microscope so I can see what they're seeing, and they keep mistaking things in the poo that for Macrorhabdus that aren't. So if you're patient and willing to keep trying, then this is something that you can do, but it's not that easy and you shouldn't get discouraged. And some people never quite get there, but most people do. Okay, Macrorhabdus doesn't just occur in infections don't just occur in budgerigars, as we've already mentioned, and they can cause disease in other species as well. And I thought I'd just, for your own interest, talk to you about what we see in Australia, and a very common presentation that we had. Let's see what time. It would be a little bit earlier in the year, was when the young galahs were coming out of the nest and the young corellas were coming out of the nest and the young king parrots were around, was a chronic wasting disease. And it was particularly common in galahs, but particularly also seen in king parrots from all over the eastern seaboard of Australia. And they would come in and they'd be all fluffed up like this one is. They would be very, very thin. They would have chronic diarrhea, and their poops would just really smell horrible. They'd have a distended proventriculus that we could see on x-rays or radiographs, or if we did a post-mortem. And sometimes they'd have other organisms that would infect their intestinal tract. There's a parasite called spironucleus that we would sometimes find. Most would have Macrorhabdus, and a lot of them would also have the beak and feather disease virus, which might suppress their immune system and make them more susceptible to some of these other infections. But they had this really severe change to their intestine, which meant that they weren't able to digest their food properly. And so we think that actually they may have had another virus infection before they got the beak and feather disease virus infection, and that they may have had another one that attacked the lining of their intestines. And so they would have multiple different things. And sadly, so far at least, we haven't really come up with a way to treat these birds and make them better, but we're trying some new treatment trials now, and hopefully we'll have better luck in the future. So another thing that you probably, or you might be aware of already, is that budgerigars, of course, are very prone to cancer, and they can get cancer of their feather follicles, and they can get cancer of their preening gland, and they can get cancer of their kidney. In particular, they get cancer of their kidney, and that presents when they are holding one of their legs up or can't move one of their legs. That's because the kidney puts pressure on their nerves. And all of that is probably related to the fact that our budgerigars are probably not genetically diverse, and that overall are just more prone to cancer. But the cancer that they get associated with Macrorhabdus does seem to be as a result of infection with Macrorhabdus. And this work has been done by some good friends of mine, and they've published recently. Mike Gardner was one of the co-authors, was on that publication. But back when I was a student working in the lab of Dr. David Graham, and so what Mike Gardner has found is that he'll see one budgerigar here, one budgerigar there that will have cancer, or will have what we call hyperplasia of the lining of the stomach, where there's more cells there than there's supposed to be, but it's still a fairly orderly progression of development, and the wall is a little bit thickened, but it's not really cancerous. But that can transform then into a cancer of the lining of the stomach, which we call a gastric adenocarcinoma. And in a sense, they're malignant locally. They're highly invasive locally. They don't spread from the stomach, but they grow right there. And these have been virtually all associated with the presence of Macrorhabdus. And we think that Macrorhabdus may secrete a substance or create a chronic irritation at the site of infection that triggers these cells to start growing. And then they grow a little bit too much, and then they grow out of control and cause this cancer. So even if a bird's not infected, not sick with Macrorhabdus, but they have lots of Macrorhabdus in their droppings, this is one reason why your veterinarian may recommend treating your bird, even if it looks healthy, if it's got lots of Macrorhabdus in the droppings, because it's possible with long-term infections, it might predispose them to developing cancer or gastric adenocarcinomas. And why it occurs in some flocks in large numbers, and in others flocks only rare individuals get it, we don't know. But again, this probably goes back to the host environment pathogen interaction. And it may be that certain birds and certain genetic lines of budgerigar are more likely to get this cancer and are more susceptible to cancer once they get infected with Macrorhabdus than others. Or it could be that there's different strains of Macrorhabdus, and certain strains of Macrorhabdus are more likely to induce cancer than others. And that's an area where new research might be done in the not too distant future. Okay, so we've talked about this organism, what it is, its history, and what kinds of diseases it causes. And now we want to get into treatment. So one of the good things about us being able to grow it is that that means that we could test for what kinds of antifungal agents will keep it from growing without having to give it to a bird. And so we could do this outside of the bird. So we call this in vitro testing, as opposed to in vivo, where that's where we're actually testing it in the bird. This is where we're testing it outside of the bird. So the way we do that is that we put the growth media that they grow in these little wells in a plate like this. And then we put a certain number of organisms in each one of these wells. And then we put the thing that we want to test at different dilutions in the wells. And then we grow them out for about 5 days. And then we look and see which wells the organism grew in. And so here we have where we see the white stuff, that's the Macrorhabdus growing quite nicely. And when we don't see any organisms, that means the Macrorhabdus has been inhibited. So we can see that in this, the column on the right-hand side, aside from the one where we made a mistake and forgot to put anything in it, that here the drug that we tested didn't work at a dilution, at a high dilution it didn't work, at a lower dilution it didn't work, at a lower dilution it didn't work, at a lower dilution. Then finally we get out here to a highly concentrated solution, and it did kill them. And so we can see that if we tested 4 different drugs here, and these 2 drugs were more likely to be effective against Macrorhabdus than these 2 drugs here, because they were able to kill it at a higher dilution or a lower concentration of the medication. And so doing these kinds of tests, we've showed that amphotericin is effective, that in vitro, outside of the bird, is effective, that Nilstat or nystatin is effective in many isolates of this organism, and that other types of drugs that I'll talk about in a minute also are effective. Two drugs that we tried was fluconazole and gentian violet, which actually people used to use to put on cuts and things. But gentian violet and fluconazole are both very effective against Macrorhabdus, are very, very toxic in budgerigars. So even though they're effective at this stage, when we start using them in the bird, we find out that they may kill a Macrorhabdus, but the birds can't tolerate them. And this work was done by a lovely young woman, Allison Bradley, who is one of my students. Anyway, so at this point in time, the drug that is used in the vast majority of cases for treatment of Macrorhabdus is a drug called amphotericin B. And amphotericin B has been used for treating fungal infections in people for a long, long time. And it's a terrible drug when given IV, like it has to be given when you're treating a fungal infection of the lungs or something like that. But it's not absorbed from the digestive tract. So we can give it orally and it lays in the digestive tract and it's pooped out the other end and it's never absorbed into the body. So it's very, very safe in budgerigars, and we can get very, very high concentrations of this without having to worry about budgerigars getting sick, because we don't give it IV like they do in people to treat systemic infections. We give it orally and it doesn't get into the rest of the body. And there's two different types that are very common. One is a thing that's called Megabac S, which is available in Australia and sometimes other parts of the world. And then there's a product called Fumagillin, which probably have spelled wrong here, sorry, which can also be put in the drinking water, and or can be administered orally. And then Nilstat or nystatin has also been shown to be effective in some instances, but in most instances, Nilstat, the Macrorhabdus will be resistant to it. So while it might be the first drug that we try because it's inexpensive and easy to administer, and we can get it just down at the chemist or the pharmacist, we might try it first, but then if we don't have success straight away, then we'll switch over to amphotericin. And there are multiple formulations, and the big problem with amphotericin is that it's fat soluble, but it's not water soluble. And so there has to be special preparations made for it to be effective. And even the water soluble forms are probably not as effective as we would like them to be. And new formulations where the amphotericin is made up into little tiny particles called micelles that might bind with Macrorhabdus or other yeasts or fungi and deliver the medication directly into them, may in the future be more effective than what we've got available to us right now. So how do we know if your bird if the treatment is working? And the first thing that may seem intuitive, but is really one of the easiest way for us to know whether the medication is working, is that they start to feel better. And then we look at repeated droppings during the treatment, and they should become, during the treatment, they should become negative. And then what we have to do to make sure that we've been successful, we need to repeat testing after we're done with treatment. And what Hamish, who did his PhD on this organism and its treatment and detection of infection, found was that it'll often beat it back, and that we won't see the organisms in the droppings But if you wait a week or two and then start looking for it again in the droppings, when we have a resistant strain, they'll come back with it a vengeance. And there'll be more organisms in the droppings after treatment than there was, a couple of weeks after treatment than there was even before we started treatment. So just because you do a single test after you've completed the treatment, if it's right afterwards or while you're treating them and it comes up negative, does not mean you've necessarily cured that bird. And you need to go ahead and test again in a week or a month later to make sure that the bird's not shedding again and the organism hasn't started growing back. And now what we're finding, and this is work that's been done in Europe and also by my student, is that up to 40% of the strains that we're testing or that we've treated inside the birds themselves are resistant to amphotericin. So the one drug that we really think is the best drug for treating this is no longer effective in many of the budgerigars that we try to treat. So what I'm going to talk about now is not a magic wand that we can necessarily will apply to your birds if they have this problem. But in the future, there is going to be some new drugs that are coming out and new formulations of these drugs. And I think that these may prove to be more valuable than what we have available to us right now. Also, we have learned that combining drugs with other things, and oddly enough, if we give vitamin C to budgerigars with Macrorhabdus in combination with amphotericin, that actually the vitamin C oxidizes the capsule of the yeast, and it makes it more susceptible to the amphotericin. And we're learning more and more about these sorts of things. So the other thing that I say to whenever I speak to budgerigar breeders is, you guys have been out there breeding budgies for the last 100 or more years, and you've gone from the green birds to birds that are all yellow and all blue and are all white and have different patterns of color. You've bred birds for the English type budgerigars that are already 2 times or 2.5 times the size of the wild birds, and you've selectively bred for this. So maybe we can selectively breed for resistance as well. And I'll talk about that in just a minute. So the thing that I want to go into now is, do we always treat birds that are infected? And if we have a sick bird, is not treating an option. So this is sort of a double negative. I'm sorry, I don't mean to do that. So if we have a sick bird, yes, we will always treat them. But if we have a healthy bird, do we want to treat them or not? And the reasons for not treating them is, if you have to catch your bird and give medication to it by mouth twice a day, then you know as well as I do is that your relationship with that bird is not going to be the same at the end of treatment as it was at the beginning. And it might be that you've really hurt your relationship with that bird. And they don't understand why you're trying to give them medication. And they might not like that at all, and they might become afraid of you. And so if you've got a nice, fat and healthy, active bird that happens to have a few Macrorhabdus in its droppings, then I would probably tell that client, I don't think we should treat that bird. I think what we should do is just monitor it. I want you to get a food scale, and I want you to have that bird hop on the food scale at least once a week, and you keep track of its weight, and we can see whether it starts to go down, make sure that it's eating, and it's looking healthy, and as long as it's eating, looking healthy, and it's not losing weight, we won't treat. We might want to check its droppings and see if the organism starts to increase over again and again, or gets higher numbers in the droppings, in which case we might want to treat. But if it stays at low numbers and the bird looks fine, then maybe we're not going to treat. But also in the back of my mind, and what I have to tell my clients, is that we also know that infection can be associated with the development of cancer. And we think the longer they're infected, the more likely it is that they might develop cancer. So again, is this a risk that you're willing to take? Not many birds come down with cancer, but some do. So are we going to have a risk that we might alienate you from your bird and treat it? Or are we going to have a risk that might develop into subsequent, have more problems later and not treat it, but we're willing to take that risk? We're willing to take that risk that it might develop cancer, but we don't want to anger your bird. So we have to make these different types of choices, and we might just think about whether we're giving the medication directly into the bird's mouth or whether we're putting it in the drinking water, and look at those different options. So those would be all things that you'd want to talk to very carefully with your veterinarians before deciding to treat your bird. Okay, so common sense. What else can we do to try to make sure that you don't end up with a budgerigar that gets sick from a Macrorhabdus infection? Well, one thing you can do is, if you're buying a new budgie, don't just go down and get it going, and actually find the breeder and make sure they're taking care of their birds the same way that you would take care of yours. That they've got a nice, clean environment, that they've got good nutrition, which I guess you guys have talked about a lot, that they don't have overcrowding of their birds, and that their birds aren't pooping in their food or in their drinking water. And if you don't like the way they're taking care of their birds, then don't buy a bird from them. Go find somebody else. Make sure that the birds that you're buying from, not only are they taking care of them well, but they look nice and healthy. And talk to them. And sometimes you can even get a recommendation from that breeder's veterinarian, and they can tell you about that. Yes, I've taken care of these people's birds for many, many years, and I've never seen them to have any problems. So the other thing that you can do is, when you bring a bird into your aviary, is always put it into isolation, where you keep them in a separate room, and you take care of them last, and you clean their cages last. If you have a sick bird, then you want to put them into a hospital environment where they've got extra warmth and they've got the best possible food available to them that they would like to eat. Put them on amphotericin and possibly some other medication for ulcers in their stomach, and treat for at least 4 weeks, and then recheck them regularly. And then if shedding returns, we may have to repeat treatment, and we may have to repeat it with another medication, or we may have to treat them for a longer period of time. If we have birds that aren't sick, we've already talked about that. You can do nothing, monitor and look at their behavior, isolate, and then treat if they develop signs, but again, that they might have an increased chance of cancer. And then if they're shedding but they look fine, then we might still want to treat them. That might make you feel better. And then again, we come back to common sense, and I want to talk about that just for a minute. So I visited a number of aviaries. I've visited many budgie aviaries over the course of my career. And I had the pleasure of visiting an aviculturist who produced some of the best showbirds that that I've ever seen, and just this incredible reputation of having beautiful birds that for show, and also beautiful healthy birds. And I went in and he had these absolutely beautiful flight cages with beautiful birds in them. And they were exercised, they were fit, just really fantastic. And he spent all this time studying the diet and making sure they got the best possible food that he could provide. And then I looked up on the shelf over in the corner of his aviary, and there were two cages there with a couple of birds in each one. And these birds looked awful. They looked sick. And I said, "What's going on over there?" Oh, he said, "A friend of mine had these birds and they were sick and he was going to euthanize them. So I took them and I brought them into my aviary. And lo and behold, they were shedding Macrorhabdus like crazy." And I'm sorry, if you've got healthy birds, you do not want to introduce a sick bird into your collection. So that's why it's so important that if you've got healthy birds, that as soon as you get a new bird, you have it vet checked, have it tested for whatever that veterinarian recommends. And you certainly don't want to put a bird that's shedding Macrorhabdus in with your other birds that don't have it. It's just not good. It's just not a good thing to do. And so we do have to use some common sense. And as much as you might want to try to save that bird, it'd be much better to put it in a home where there aren't any other birds than it would be to put it into your collection and risk that your other birds might come down with whatever it had. Okay, well, at this point in my talk, some people get kind of depressed and think, "Oh my God, should I just go shoot myself? Is there nothing we can do?" But there are some things that we can do. And there is some hope about Macrorhabdus, I think, in the future. And I've already mentioned it, that we've got new treatments that are coming along. And that I think that these will help us to treat birds. We've got new antifungals that are coming along and new formulations, and I think we'll be successful with that. But I think we have other options, which in the long run will make it so that fewer budgerigars suffer, fewer people have to deal with sick budgies. And that is to take a lesson from the poultry industry and to breed for pathogen-free flocks of birds. We know that some birds are more likely to get infected and some birds are more likely to get sick than others. And so if we could get a one individual breeder whose whole goal in life was to breed for birds that were resistant to infection and had birds tested, males and females tested, that were negative and bred those and only those, and looked at the young and saw that they were free of disease and bred those and looked at young and saw that they were free of disease, then eventually we could, I think, select for birds that were no longer susceptible to infection. And then we would build up a whole big population of those that could be sold as pets. And we could ask more money for those because they were ones that wouldn't have these health problems necessarily. And then if you really want to have some fun, when you want to produce some Macrorhabdus-free budgies, go ahead and incubator hatch some eggs from budgerigars. Let the hens incubate them up till just about the time that they're ready to hatch them. Pull them out and put them in an incubator. Hatch them out and then hand rear them separate from all the other birds that might have infection. And those birds will be free of Macrorhabdus. They'll be free of all other kinds of virus infections and all other kinds of things. And boy, I'll tell you, you haven't met an amazing little budgie until you've met one that's been hand raised. And I had students do this, and I would come into the room, the aviary where these birds were, and they'd have these baby budgies climbing all over the top of them, on top of their heads, up and down their arms, up around, climbing up their fronts and down their backs. And they were just the most wonderful, friendly, lovely birds. And so it's not easy to hand raise a budgie. But it's not that hard after you get used to doing it, and they will be disease-free of many diseases that they might otherwise get, and they'll be the most friendly and wonderful little birds you could possibly imagine. Okay. So again, I think that we could definitely, by selecting for parents that raised young that didn't have Macrorhabdus, end up with Macrorhabdus resistant birds that would be much more healthy. And we could selectively breed them for birds that were resistant to polyomavirus. We could selectively breed them for birds that were resistant to beak and feather and yeast infections. And selectively breed for birds that were less likely to get cancer. I think that we could make some lines of budgies that would just make absolutely wonderful pets. And I encourage budgerigar breeders to do this. And I know that they can. So thank you very much for your attention. And no, this isn't a budgerigar. This is a little azure kingfisher that lived in the Nepean River that I used to kayak on. And you knew they were around because you could hear plop as they dropped into the water and caught a fish or an insect, and then turn around and look, and you'd realize that you'd paddle just by one when you're in your kayak. They're amazing little birds too, feisty little birds that kind of remind me of budgerigars. So thank you for your attention. And Amy, I guess my question will be, do we want to take another quick break before we start into questions, or start into questions and then take a break? What do you think?
Yeah. Do you want a quick break? We'll give everyone maybe like 5 minutes that we get to rest your voice a little bit, and then we'll hop back in.
All right, that'll be great. Let's do that.
Okay, sounds good. So I feel like that was 5 minutes, right? So
I
I've
think so.
collected everyone's questions. So if you guys want to turn your videos on, so you can, I'll kind of read off the question, but if you guys want to add anything while I ask, we can do that. So let's start with the first question. So Nathan submitted a question first. So Nathan asked that he wanted to ask about the determination of amphotericin B prep and administration. It was really good to hear in your slides that there are multiple formulations, because we've kind of noticed different reports and experiences from different people about the formulation of the drug. Looks like in a 2014 review, you had both a 100 milligram per kilogram dose and a 25 milligram per kilogram dose as options. And also, do you have any suggestions on the volume? We've noticed that for my vet, we can get something like 0.02 cc's or 0.03 cc's, but we've also seen prescriptions that were like 0.1 cc's or 0.2 cc's, and they were very difficult to dose. So I don't know if you have any suggestions around that. Yeah.
Right. So obviously the amount that you have to give to get the effective concentration is going to depend on the concentration of the medication in what you're giving.
Mm-hmm.
So it's not so much about the volume as the amount.
Mm-hmm.
And veterinarians have a couple of options on making up these formulas. And one of the problems with Nilstat, one of the reasons we don't use Nilstat, for instance, is that's not amphotericin, but it's Nilstat. But you have to use a large dose when you give it. So that's one of the reasons it's not used very much. Mm-hmm. But a good avian veterinarian will have a good compounding pharmacist that they'll work with. And the compounding pharmacist will be able to make as high a concentration as possible, so the volume is as low as possible. And I think what we've found is that there's been dosages that have been offered as being effective, and they've gone from 10 milligrams per kilogram to 25 milligrams to 100 milligrams per kilogram. And I think what we're seeing is gradual resistance developing in the Macrorhabdus. And so in the past, we might have been able to use lower concentrations for it to be effective. And then now we probably have to use higher concentrations in some instances, at least.
Yeah.
Because like I said, about 40% of our isolates, the ones that we looked at anyway in Australia, were resistant to amphotericin, and it would knock down the numbers that were being shed, but it would bounce right back after we completed the treatment.
Yeah, that's a really good point. We've noticed that. Nathan and I have discussed this because I had a few cases of birds coming in. They had very low numbers. We decided to treat because I could handle the behavioral fallout, and we treated, it cleared. And even years later, I asked them on necropsy to specifically look and do histopath to look for the organism, and they did not find it. So my experience originally with this organism was a lot more relaxed. But then when Nathan's cases came up, it was exactly like you described, like a resistant strain. We treat and again, it comes back, and we treat and again, it seems to get worse. I don't know if you've seen any particular strains that seem to be worse, or it's just kind of like a general trend.
Well, we aren't at this point in time able to characterize the strains particularly well. In people, what they do is they actually sequence the entire DNA sequence of a fungus or a bacteria or whatever, and then look for certain mutations or virulence factors. And we haven't gotten that sophisticated with Macrorhabdus at this point in time. I think so. Just to give you an idea, this is something that has bothered me all my life, and I won't get into it a great deal, but it's very, very common for some budgerigar breeders to treat their birds every week or every month for one thing or another, or multiple drugs now and then that again and again. And when you do that, especially if you don't even have the organism present in that animal, what we always say as veterinarians, you don't treat an animal unless you can prove that it's got a problem in most instances. And so there's in a lot of breeders, especially the ones that breed for show, and I'll probably get crucified for saying this, I don't want to, but it's just a normal thing for them to use tetracyclines and amphotericin and other drugs over and over and over again, treat for worms over and over and over again. And eventually, if we do that, those organisms will become resistant to the drugs that we're using.
Yeah.
And so that's where we can run into problems. And I think that's maybe one of the reasons why we're seeing more and more drug resistance. Now, in your instance, Nathan, what I would suggest is that you either treat with some vitamin C in conjunction with the amphotericin and see if that helps. Vitamin C is not anything that's going to cause any problems. And I can send Amy the drug dosages that we would normally do, and she can pass them on to you. And so you can see whether or not that may help.
Yeah, that's great. Next question Nathan had was, your general experience working with amphotericin B. Yes, it's poor water solubility, and any up and coming work on lipid emulsifications or new formulations.
Well, there is. I don't know of it specifically. My graduate student, Hamish Barron, wanted to walk away from his PhD with something that he could give to every breeder out there that was going to solve the problem. And I know he's still thinking about that. And I will email him and ask him to forward me anything that he's got his ear to the ground and more in the grapevine than I am. So I can send off any new information, but I'm not familiar with that. Again, it might be one of those things where you're if you've been given it orally, you might want to try water soluble that you can actually put in the drinking water, and just keep it on it for a long, long time, like 3 months or something like that. That might be another option, but I would start with vitamin C and then see what happens after that.
Yeah, that's great advice. Um, do you know if I'll interject a question into Nathan's questions that are kind of related. Um, do you usually expect to see side effects with amphotericin B treatment? I've seen some kind of conflicting things. I never saw any when I treated my birds, and I think they were on a pretty high dose, but I've seen some like mixed reports.
Well, it has not been my experience. Now, if you go and look up amphotericin B on the web, not in relationship to budgerigars, but for people or whatever, it's the last resort drug that's given IV to people that have very, very severe infections because they're immunosuppressed or they've had a catheter in for a long period of time, or they've got a really nasty fungus like cryptococcus or aspergillus or something down in their lungs. And the nickname for amphotericin in the hospital is amphoterribilis, and it makes people really, really sick. It makes them feel horrible when it's given IV.
Yeah.
When given orally, because it's not absorbed from the digestive tract, I have not seen any indication. I've not seen any complications with it.
Hmm. Um, could I ask a slight clarification on what you mentioned before about the amount versus the dosage? So is it that we're looking for a total milligram amount given on the body weight of the bird?
Right.
Mm, got it.
And one thing that I forgot to mention and that I will mention now because it's really important is that when we did that kind of testing, which called the in vitro testing, which I showed you those wells where they either got cloudy if the organism grew or were clear if the medication worked, we found that these what they're called low toxic antifungal medications, which is sodium sodium and potassium sorbate and
Oh, the benzoates.
benzoate. Yes,
Yeah.
yeah. And we got really excited about that, and we tried some treatments, and it looked like it was working. But I do not recommend that under any circumstances now, because believe it or not, budgies do drink water. And when they drink that, they get too much sodium and they get too much potassium, and they actually get sodium and potassium toxicity potentially. So we do not recommend using that.
Yeah, that's really good to know. I've had clients from the UK tell me that they're not able to source amphotericin B and this is what they're being prescribed. And I know that paper, I've seen that paper where, especially for the birds who were inbreeding, they were drinking a lot more water. The side effects were even more severe and the fatality level was even higher. Um, but it seems in some places in the world, I don't know why, there's some kind of supply chain issue where they're not able to get it. Yeah, so I'm still seeing places where it's popping up, unfortunately. Yeah.
Well, I think even in those situations, I think if you can work with a compounding pharmacist,
Mhm.
that in most places in the world they can get amphotericin.
Mhm.
So that would be the first question I'd ask my veterinarian is, are you working with a compound pharmacist?
Mhm. Yeah.
And have they said they can't get it? Well, maybe you could call one of your friends or find somebody else that's doing this, and they might be able to. They might know another compounding pharmacist
Mm.
that that has a better source somewhere.
Yeah, for sure. Um, Another question, it says, what were your observations regarding shedding with fecal cytology and PCR? Is the shedding really inconsistent, like the intermittent shedding? And how frequently do you see difficulty in detection, either because it's too low in PCR or because of human error? Because with Nathan's case, we did multiple PCRs and different types of fecal tests. None of the birds came back positive, but of course they began to progress. And one bird who did pass away, we did do a necropsy, and it was pretty definitive that it was Macrorhabdus. So, and the adenocarcinoma at the same time. And the birds were a couple years old, I think, right, Nathan? Remind me how, yeah, 2 years old. Yeah.
Okay. Well,
Yeah.
one time I had a lovebird that had Macrorhabdus, and we treated it, and Macrorhabdus disappeared in its feces, and it continued to go down and go downhill, and finally it died. And when we did the postmortem, we found that it had cancer, but the Macrorhabdus was gone.
Yeah.
So in that instance, we thought we just weren't finding it, but in fact, we had gotten rid of it, but that has caused the cancer, so that the cancer was there making signs that looked like Macrorhabdus, but the Macrorhabdus was gone. And we know, and if you read some of Hamish Barron's papers, he's got all that data in there. We know that that you can have lots of infected birds that aren't shedding. And that's one of the frustrating things. And he found that the suspension technique, where he mixes it with saline and then just takes a drop off the top of the mixture right straight away, and looks at that, and PCR, that they're about equally sensitive.
That's very good to know. Yeah. Yeah, I've noticed that at least here in the US, I've seen quite a few situations where we submit an obviously positive sample and get some results back from the lab that are negative. I mean, I spend most of my time working on the bench, so to me, it can only just be poor lab technique. I don't know what that is.
Well, I'm not going to say this out loud, but I might say it anyway. There are certain labs that I have more confidence in than others.
I agree.
And so that would be one of the things that you'd want to talk to your veterinarians about, maybe see if he could, post on BirdMed or something like that, and get some feedback if what they're getting is not doesn't match up with what we're seeing in the bird.
Yeah, that makes sense. Yeah, we've seen with Nathan's case, probably in my bird as well. My budgie that passed away back in December was the same case. Around 3 or 4 years ago, we saw a very low amount of shedding. We did decide to treat, negative, and she progressed all the way until later. She actually, from another bird that came in unknowingly, I didn't know that bird had mycobacteria, so that was a different thing that ultimately caused me to have to euthanize her. But I asked them to also look, and she no longer had any presence of the Macrorhabdus organism, but she did have the cancer. So it was the same thing. Yeah. And now we're wondering with one of others, Nathan's birds, we've treated him multiple times. We don't see shedding. It's hard to tell, but we are still seeing symptoms. We're wondering if it's just that these residual symptoms of the cancer or something else is still there. So it makes a lot of sense.
The only other thing to speak to, Nathan, is that, and it's probably not the case, but as I mentioned in my talk, we can see that they can have other things at the same time. So in Australia, it's particularly common that they'll have trichomonas infections. And they'll come in looking very much like Macrorhabdus, and then we do a crop wash. They find these little swimming guys all over the place. Treat them for trichomonas, which, by the way, is much easier than Macrorhabdus, and they get better. So again, there are other things that can cause this, but one thing that potentially can is that you've got a tumor starting to develop.
Yeah, that makes sense. Next question is, do you have any difficulty with post-mortem detection in M. ornithogaster due to the amphotericin treatment? or was post-mortem observation mostly clinical signs like thickened mucosa or ulcers. Um, he said
Well,
for his samples, they were not positive for the Macrorhabdus, but they did find the adenocarcinoma. Mm.
yeah. So what Hamish found and what surprised me a little bit was that the best that the most sensitive test for infection with Macrorhabdus is doing a scraping of the isthmus and doing his suspension technique.
Mhm.
And that was even more sensitive than looking at the tissues under the microscope.
Mm.
And that's because in order to look at the tissues under the microscope, you have to be very, very careful in the way that you select the tissues, that you get that junction. And
Mm.
it's fairly easy between selecting the tissues and the histopathology labs, putting them in the paraffin and making the sections that get exactly at the right part.
Yeah, that makes sense.
So if you can scrape the proventriculus and the jump at the isthmus there, that is overall exactly the most sensitive. And if you scrape that and send that in for PCR, they're equally sensitive.
Mhm. Um, yeah, it seems like there are specific comorbidities that appear to be gastrointestinal. Do you have any guidance on possible symptom management during the amphotericin B treatment? We've seen some improvement with meloxidil treatment. So and there was some a suspected pneumonia that enrofloxacin was used to treat called so yeah, and then the crop enlargement and the frequent neck adjusting that looks like vomiting was the most difficult, but it seems to get better with some kind of anti-inflammatory for some reason. Um, yeah.
Well, I guess the first thing I would say is that if we go back and think about what's happening in that stomach with the Macrorhabdus infection, we've quite often have ulceration of the ventriculus, the coiling lining, and also the proventriculus. And so any bird that's sick, I always will treat with carafate, which is sucralfate, and
Mhm.
that will bind to the lining of the stomach and help it to heal up faster. Using an antacid is also another thing that might help those birds. And quite often there's mixed infections in the stomach, and you might have an overgrowth of bacteria as well. And so sometimes providing an antibiotic can be helpful. So that's one of the things that I should mention that I intended to mention. One of the things that you'll read is that you should treat with apple cider vinegar to change the pH of the stomach. Well, guess what? Macrorhabdus likes a nice acid stomach. So that work was done. And again, I won't comment on it particularly, but some of the testing was done with just pH papers on the surface, and I'm not sure. Anyway, I don't have any feeling that using apple cider vinegar is necessarily going to help treat
Yeah.
macrorhabdus.
Kind of along the same vein, amongst the growth conditions, they seem to like to use sugar as a fuel. We do see a lot of new products come on the market that do add simple sugars to the products. Do you think there's any risk of, let's say, using those types of products that might increase the risk? There's no need for the birds to have those simple sugars in their diet. It's mostly for palatability, for conversion, which we can do without that if we just know the behavior well. Um, but I'm kind of wondering if that's fueling the problem.
Well, we know that with Candida, in all species, pretty much, if you give them a high sugar diet, especially in lorikeets, we know that if we put them on the standard lorikeet mix,
Mhm.
that they'll get Candida overgrowth of their digestive tract almost straight away. So
Mm.
there is a possibility that could help Macrorhabdus to grow.
Mm. Yeah, I don't know too much about the physiology of the digestion there. It's something like um, grains and things like that, which are, starches that would then get broken down to be sugars. Is that mostly in birds sequestered to the small intestine, or some of that happening higher in the digestive tract?
Well, none of it's being absorbed there.
Hmm.
They're really, I mean, their digestive process is very similar to simple stomach animal, where they're producing pepsinogen and acid initially, and then that breaks it down to a fine grain. There's no absorption of anything that occurs in the stomach, and
Yeah.
then it would go into the duodenum. Now
Hmm.
there is some reflux from the duodenum back up
Hmm.
into the stomach, so there may be some digested material that gets refluxed back up into there. But most of the absorption, essentially all of the absorption, is going to occur in the small bowel.
Hmm. And is there something particular about the isthmus that makes this such a vulnerable area for infection for this organism?
Well, there is, but I don't know what it is.
There's something
What I
about
can
it.
tell you microscopically is that's the transition between the proventricular glands to where we're producing the coilin.
Mm.
And so if there is coilin there, it's very thin, and it's not as tough as the stuff in the gizzard per se. I suspect it's the pH is exactly right. And also I think that that it probably it allows the Macrorhabdus to go down in between the glands there,
Mhm.
and that the Macrorhabdus does clearly feed on some of the secretions that are being produced by the host. So that's probably why.
Yeah, that makes sense. Um, is there any connection that you see to breeding, a breeding state or breeding mode? One, because for a lot of owners, we are frequently trying to keep our budgies from constantly laying eggs. Kind of going to your point about selective breeding, the huge disparity that we're seeing amongst budgies, and a lot of budgies coming into breeding with very little trigger. This is a little bit of a rant, but so many budgies are coming into breeding, they get quote quote chronic egg laying, which we can solve, but it's just these budgies shouldn't be breeding when there's so few triggers in place, so few factors in place. So I'm suspecting that people are not being careful with their breeding. So a lot of us are getting birds with that situation, where a tiny increase in the metabolizable energy and they will not stop laying eggs unless you fix that diet, which is quite interesting. But do you see any connection with other life states that may make them more susceptible to the organism besides like another disease, I guess?
Okay, well, I guess two points. One is to why we have cockatiels and budgies that are so prone to breeding is that breeders are selecting for those birds too.
Yeah, I've always suspected that this is the case, but I've never been able to prove it or bring any evidence other than just I can see it happening with my eyes that people have birds who are, I see breeders all the time. They go, oh, this bird gives me big clutches. Love this bird. So they keep breeding that bird and that bird goes home to another home. Or it's the dead of winter and the temperature is low, but that cockatiel is breeding and they're continuing to breed that bird. And then the ones that don't breed are the ones that get culled, quote, quote,
Yes.
accidentally. And it's just getting worse and worse and worse. Um, that's good. At least I feel like I'm not just crazy.
Well, many years ago, I had worked with a cockatiel breeder, and I went over to his place, and I said, "What are those birds over there? Are they the ones that aren't giving me as many eggs, that aren't breeding as often?" I put them together. I put the male and the female together, and if they don't go at it, then I find some other to pair up.
Yeah.
So at least in that instance, and I suspected probably in others, humans rarely exhibit one behavior that other humans haven't exhibited already. So
Yeah.
I think that they do. They select for birds that give bigger clutches, that are more likely to feed their young, they're more likely to produce offspring, or more likely to go into the nest box straight away.
Yeah.
Okay, so that's my informal scientific perspective on that, which has absolutely no value, except it might be true. So what we know is, as I think I've mentioned in this talk already, is that those really young budgies that are 10 days old in the aviaries, we've got widespread infection. At least in my group of birds that I had this 100 breeding pairs that were for research. And for our nutrition trial, when we did postmortems on birds at 10 days of age, virtually 100% infected. When we did it 25 days of age, virtually none. So something in their stomach, as they transition from that, maybe it's because they're getting food that parents have regurgitated and partially digested or something already or something, or maybe the environment in their stomachs changes. I don't know what it is. But the younger birds were more likely to be infected than the near fledgling birds. But having said that, there was this one female. She's a beautiful Lutino hen. She produced massive numbers of chicks, and every single one of them was shedding the organism, even out to 25 days. So there's something different about that one. And that's why I think that we could probably breed for resistance to this.
It's interesting you mentioned that, because one of the observations from Nathan's case was he had radiographs done, right? And one of the things we saw was a very enlarged testes in one of the birds, which in a lot of birds, they come in, they have breeding triggers all over the home. Usually, if we withdraw those breeding triggers, we'll see the behaviors kind of pass and it tends to get resolved in most cases. But here it didn't matter. Those testes remained enlarged despite being there were essentially no other breeding triggers other than the two male budgies, and he was also a male. So it's interesting. I wonder if there's some kind of something going on there. If you've ever seen that the disease is linked to high breeding condition hormones. I don't know. It was
Well,
one of
those
the
budgies are going to
strangest.
be feeding each other back and forth, whether they're male or female, and regurgitant feeding. So that would be a good way to pass the disease back and forth.
Oh, that could be, yeah, that because they have the high hormones, people are selecting for that. They do more of the regurgitating behavior, and naturally they end up being more infectious as a result of that behavior. It's possible. Yeah.
So we think that there's probably two ways that the organism is transmitted between birds. And one is regurgitant feeding, and we think that that's probably how the parents feed the chicks. But also we know, because I did these infection trials in chickens, day-old chicks, that we take half our chicks and don't infect them, and take half our chicks and infect them, and then 5 days later check, and they're all infected. So, and the day-old chickens would never think about feeding each other. It's just the fecal contamination of the ground, and then they clean themselves off or get their food contaminated, and then that's how they get infected.
Yeah, that makes sense.
And a lot of these budgie nest boxes are at least in some instances, especially the commercial aviaries, they're just stacked full of feces in there. So there would be a huge chance for contamination. But we would always want to, again, think about the environment at home and make sure they're not pooping in their water, they're not pooping in in their food.
Yeah.
And I guess if you've got birds that are feeding each other, you don't want to just treat the one that's shedding, you probably want to treat both of them
No. Um,
or
oh, question.
separate them during the process.
Yeah, that was what we did. So the birds lived three birds, three cages, and they were not never allowed to feed each other after that point, essentially, because there was no other way to control. Yeah, but it was very tricky for sure. Um, Question from Sammy, is there any research done using vaccinations maybe as a method of treatment?
Well, that's a really good question. And it's one that I don't think that anybody's done. Now, because we were able to grow Macrorhabdus in such large amounts, a friend of mine and a colleague in Italy, his name is, I think it's Giacconi Rossi, Rossi. Don't know how to pronounce his first name, and I forget the spelling. But he asked for Macrorhabdus so that he could test to see whether they had antibodies against it.
Mhm.
And I don't know what he found, but you might see if he's done any research on that. And of course, vaccine production is a challenging thing to do. And ideally, what we'd want to use is just a specific antigen and not necessarily the whole organism.
Yeah.
But we can grow the organism, we can kill it so it's no longer infective. And we might be able to inject that under the skin and see if we induced immune response. So I think that's a reasonable approach, and that maybe someday somebody will do that. I'll suggest that to my graduate student, Hamish.
Yeah, someone should definitely, the more we talk about the science, the more I think all these projects that
Yes.
we have to do, they're truly endless. Let's see. Yeah, so another question from Nathan, have we ever considered administration of amphotericin B via some kind of food vehicle as opposed to handling, or is there any reason that might not work? Sometimes for a lot of us, if we can't restrain the bird that easily, I've had some success putting it on a piece of millet or on a tiny bit of cooked grain that's a high value food, and they will eat it that way. I don't know if you've seen any other success with that.
I haven't done that. Again, Hamish, who wanted to cure this thing and make the perfect diagnostic test and everything else, sort of ran out of time at the end. And one of his goals was to look at the possibility of creating a micelle with a macro with the amphotericin in it, so that would get to the area that we wanted it to get to. Or what he was actually looking at was the possibility of creating a small latex bead that had the amphotericin in it, and that the latex bead would be essentially like grit, and it would sit in the gizzard, and as it was worn down, the amphotericin would be released to the environment. But we never, and at a slow, constant rate, but we never got to that point. So I think there's a lot of possibilities out there that might someday be able to be tried.
Yeah. And if, kind of like you mentioned, sometimes we'll treat with amphotericin B and then it will come back with a vengeance. Maybe we treat again, maybe for higher dose or a longer time. I assume it's something that we can't use indefinitely. At some point, it just stops working. It sounds like there's not a toxicity issue at 100 milligram per kilogram, which is already high.
Yeah, I mean, that's an enormous amount of amphotericin. There's no question about it. And that's as high as we've ever gone. But I don't think, because again, it's not absorbed. It's only in the digestive tract. and it's going to kill Macrorhabdus. It's certainly going to kill Candida, and will kill other yeasts that might be in their digestive tract that potentially could be a pathogen. And I don't see any problem with giving it. But I think what's going to happen is that when we start to see resistance, what we're doing is selecting for a strain of resistance, and that's not good. So if we keep treating, then we're probably, in the long run, going to make amphotericin less and less effective.
Yeah. Is there a point at which, let's say you've treated it 3 times or 4 times, is there a point at which it's just becoming so resistant that it's difficult to keep moving forward and we have to consider perhaps less ideal options?
Yeah, I can't say that I've ever been in that situation where I've repeated over and over and over and over again. Again, I can run that by Hamish and see what his opinion is on it. In fact, I'll contact him and see if he wants to answer some of these questions or what he thinks.
Yeah. I think you mentioned already that we don't know too much about strain characterization. But do you see any correlation with certain strains having more severe symptoms, or do you think it's more a factor of maybe the bird itself, like the factors of the host, maybe the bird is more inbred and
Well, I'm confident that I think that there are strains that are more likely to cause this cancer type situation. And I do feel that I don't know, I can't say for sure whether there are strains that are more pathogenic, but I would not be the least bit surprised if there were.
Yeah. Yeah, that wouldn't shock me either, because I think just the difference in experience between me and various other people who've had this to deal with this disease, it's so different. Even it's just so dramatic at the difference in presentation. So yeah, I wonder, it wouldn't shock me either if, and especially always they're geographically separate. So they're geographically in different places. So I'd be curious to see like if there's one particular area where that version of that strain is coming from one pet store, one breeder in that geographical area, and that's the strain that's there. I don't know. It's possible.
Well, I mean, it may be the strain, or again, it may be the genetics. So though that would be hard to parse out, but again, I guess if I had another life to live and money wasn't an issue, I'd go out to West Texas somewhere where the environment was much like it is Central Australia is, and I'd have my aviaries outside, and I would start breeding my budgies for resistance. And then I'd, as they say in Australia, sell them and make a matzah. No, I wouldn't. But,
I have a long thought now that I've seen, this huge disparity. And originally I thought I was kind of just imagining things. I didn't want to, jump to conclusions, but I found a paper back from 1970 from, I think, Barbara Brockway and Edmund Wyndham, and they compared the gonadal responses in wild budgies compared to the captive ones. And I think this was maybe in like the 1950s. So at that point, they'd already been in captivity nearly 100 years. And they also documented that big disparity. And I was like, okay, so I'm not the only one who's kind of noticing this, that different birds seem to be really, really different in their hormonal response, despite being on the same diet, the same triggers, the same temperature, the same humidity. But I thought that too. I thought if I started breeding budgies, I'd start breeding for birds who only breed when all the boxes are checked.
yeah.
Yeah, when all 6 boxes are checked, then you can go. Because even, yeah, like Nicole, who's on the call, she has 10 budgies in her home. And we see the disparity from one side to the other. She has one female named Sugar who seems just cool as a cucumber. She's like, breeding? What's that? Despite having 4 males in the house. We keep the die under control and everything, but then on the other side, there's another budgie gray who is not even 6 months of age, and she said, "It's go time. I'm 6 months old and I'm ready."
Mm.
So, and she cannot handle any even minor increase in metabolizable energy. She will drop an entire clutch of eggs with 3 days warning. Like it's crazy. Yeah. Um, yeah, we only see a few days. We see, oh, we're noticing the outward behavior, and then we go, oh no, like something's going on there. Um, so yeah, if I too had a breeding, if I had a breeding effort, that's probably what I would breed for. Because I too, yeah, I don't care about the colors and the size so much. I just want them to be healthy. Goodness. Um,
Yeah, can you imagine if we had a budgies that could live to be 12 or 14 years old or something like that and didn't
yeah.
get cancer and everything else? I mean,
Yeah, for sure. I think people are, and it's so often that I don't think most breeders even know. I mean, the style of breeder we had decades ago where people are really intense about it, and these hobby breeders that are like, I'm an aviculturist, I'm this is my thing, that has kind of decreased because people are they care about their birds, they love their birds, they don't want to keep adding to the population. Now these big production breeders have come into the picture, at least here in the US, where it's very frequently people come to me, they're like, Amy, I want to get a budgie from a good breeder. And it's so difficult to find someone who's doing it right. It's just near impossible. Even when people come to me before they have the bird and they do everything in their power, we can't even find one who can check all the right boxes. And we just have to settle for one thing or another, or settle for the pet store where we know that that's, not ideal.
mm,
Mm. Yeah. Because here we have, yeah, huge production, the giant productions, and I know there's not just the Macrorhabdus, but the mycobacteria that will pop up there. Ooh, that's yeah,
yeah.
yeah. Um, do we know if there's a reason why Macrorhabdus is more common in some bird species than others? Um, is there something about budgies that
Mm.
Hmm,
Well, the ones that seem to have the most trouble are seed eaters, predominantly the finches, the budgies, the lovebirds, cockatiels. Whether that has anything to do with it, whether that creates a good nutrient-rich environment in them, I've never seen it. I've never seen it in a pigeon. Never seen. I mean, for instance, there's another example. Parrotlets, for some reason, are incredibly susceptible to Macrorhabdus. And what makes them that way, I have no idea. So
hmm.
there's a reason, but I haven't figured it out yet. I left that for Hamish. That's his job.
Yeah, the science is out there. We'll find it one day.
Yeah.
Um,
I mean, we certainly know that there's certain species of parrots in Australia that are quite susceptible to it, and other ones we never see it in.
hmm,
I've seen it once in a rainbow lorikeet, but never more than that.
interesting. Yeah. Something about maybe the digestive environment there. Maybe the environment is just not right.
Yeah.
Yeah. Um, let's see. It says, oh, do we know anything about the genetics that might make them more susceptible? Or this is just something we're gonna have to select for and we don't know.
Yeah, well, I mean, that would be an easy project for a veterinary student to do.
Mm,
If they could just go back into Mike Gardner's database or the database at Tufts or at the Animal Medical Center or something like that and say, uh, is there a color phase, a color type that is more likely to present with Macrorhabdus?
mm,
I can remember back in the day, back in the 90s, when they were starting to breed different color phases of cockatiels and things. And we used to talk about how Lutinos were really susceptible to diseases in general.
Mm.
But then they started to backbreed, and they were just highly inbred. And that's what made them. And once they started doing back crosses and things, then that no longer was an issue.
Yeah. I wonder if it could be done by to look for specific genes that are not visible. It'd have to be something like a GWAS, like massive data set of like whole genome sequencing. Now that we have DNA sequencing available compared to decades ago, um, it'd have to be a whole genome sequencing and see, over 10,000 birds who do and don't have disease. Is there some gene that pops out above the threshold? Um, I wonder. It's probably
Yeah,
more
I mean, there's
complex.
no doubt that's becoming increasingly possible.
Yeah.
Being done in dogs all the time, and cats, and again, it just boils down to the industry and wanting. I mean, so I can remember back in the day when we were bringing a lot of birds in from the wild, which in this day and age I would be reluctant to ever do again and feel horrible that we did what we did when we did it. But the aviculturalists started putting money into this, and they were the ones that led to people like Branson Ritchie being able to identify beacon feather disease, and helped my polyomavirus research and our herpes virus research. And I think in the end, our bird breeders and pet bird people are going to be the ones that are going to need to drive this.
It's interesting you say that because I had the same thought. I noticed it's very hard sometimes in academic institutions, depending on where funding is, what kind of efforts want to be funded. Um, Research is expensive. It's an expensive effort, both in time and in money and supplies. Um, but I really thought about that too. For this community, as it continues to grow, we're already kind of right now, we're just doing a lot of like collecting case studies. I'm already starting to see trends pop out just from the behavior side, different kind of behavioral interventions that we can do. But it'd be really interesting as it grows if it can eventually be able to support some kind of research effort. And it doesn't have to be huge to start. It can start with something very small. But I feel like I agree. I've always thought it had to come from the people who are interested in improving the situation. Yeah, perhaps this community can be
Well,
that one day.
the budgerigar breeders that have bred for these show animals, they know all the genetics.
Yeah.
They know all the history of their birds. So it might be as simple as going into somebody's aviary, looking at which ones are shedding, which ones aren't, and saying, okay, let's look at the genetics of these for the last 5 generations or 10 generations. And I bet a lot of budgie breeders would have that information.
Interesting. Definitely worth noting. Yeah. Um, let's see. Yeah, you mentioned that the chicks contracted from their parents. Some will be infected at time of weaning and others will not. So is it that some chicks were exposed but never actually contracted it, or all the chicks contracted it, but only some show symptoms and others don't show symptoms?
Well, what we know is that at 10 days old, in my aviary anyway, that pretty much all of the chicks had it. But at 25 days old, the same chicks from the same clutch didn't.
Mm-hmm.
So they were infected, but something changed in their digestive tract that no longer supported the Macrorhabdus.
Mm-hmm. Yeah. I keep coming back to this diet thing because it really makes me wonder, because that also kind of makes me wonder with the diet. A lot of these newer pelleted diets, they are extruded, and I wonder if that high digestibility could be an issue because one of the things I saw one publication where they did a dye conversion of budgies from seeds to pellets, track some different things. And the one thing that they did document was a big spike in the Macrorhabdus shedding, um, which wouldn't surprise me. But I've noticed that sometimes with certain types of birds, I wonder if it's something about a highly digestible diet, which is what the babies were receiving until they started eating solid food. Now they're eating seed that is not digested at the level of the crop, not easily, at least, probably. Um, or at least like by the time it gets to, I really have to brush up on my digestive physiology on where like the amylase is, if there's amylase before the proventriculus or not. I
Well, there'll
guess
be
that's
amylase in the saliva.
so there is going to be some digestion of the starches. So I wonder, yeah, if it's, you know, a whole seed, it's not broken down as fast as an extruded pellet. That's what happened with my male budgie. Was I moved him from Zupreme, which is actually a much higher sugar pellet, to Harrison's. And I think that diet change was a bit too fast because he had no problem just eating whatever was put in front of him. And that's when I got the big flare. And of course, the huge inflammation of the crop on the left side. Same thing. He was negative for that. We treated with amphotericin B, completely cleared. But the crop inflammation would not go down. And that was what ultimately, I think, potentially progressed to what ultimately became a very acute cardiac event. Like he one day just went from healthy to myocardial breakdown. Like, and that was what we saw in the necropsy. So I always come back to this diet thing that I think high sugar diets might be
Well,
an
I
issue.
think I agree with you. I think that if we, whatever it is that could change the environment in the stomach, whatever that was, I think that there's got to be something that would make a difference. And whether it's volatile fatty acids or availability of carbohydrates or protein or what, I don't know.
Yeah, but potentially worth more research. Um, and can a bird show symptoms or pass away even when not shedding? Or will we always get some kind of, by the time they pass away, we would have seen other symptoms?
Well, at least in my experience, the vast majority of birds that die with Macrorhabdus have lots and lots of organisms in their stomach and are shedding, usually lots, but not always. Now, what could that mean? Could that mean that we've gotten rid of it and it's transitioned and now we've got a cancer in there, or that there's been damage done already and then that causes them to die, or they start to bleed from an ulcer or something like that. It's, you know, it's difficult to know.
Yeah, and when we see symptoms in a bird, does it tend to be all or most of the symptoms, or does it depend? The undigested seed, sometimes I feel like it pops up a little later. So sometimes people will bring me a budgie that looks sick, or, they show me a photo. Amy, do you think this budgie is sick? And they have the very classic, very hunched posture and they look quite ill. And I say, okay, this bird is clearly sick. I'd recommend you take them to the veterinarian. And sometimes a little bit later, I'm like, watch their droppings, as you might see undigested seed in the droppings. And usually, boom, there it is a couple days later. So I don't know if there's some kind of order, or most of them all tend to show up at the same time.
Well, undigested seeds mean that the ventriculus is not doing its job. The grinding stomach's not doing its job. I mean, it's pathognomonic for that problem. And to get extensive ventricular disease, or to have it not grinding because there's a lot of pain or anything, that would make sense to me that might be in a more advanced situation.
Yeah. Let's see. And how quick are the rebounds that we see after treatment, especially with the suspected resistant strains, or do we know the general incubation time? It seems like, yeah, like birds that are 2-3 years old seem to have some kind of situation. Could we call that an incubation time?
I wouldn't. I don't think it is. I think that I think that an incubation time would technically between, well, it could depend on what your definition of whether we're talking about clinical signs or whether we're talking about colonization of the gut and or enough of organisms there to, so it's a bit a matter of semantics, but we know, and I don't remember exactly, but when we infected our chicks, it depended on what dose we gave them, the chickens. But they, within a week or 2 weeks, they had heavy colonization of their digestive tract. In Hamish's paper, which I don't recall right off the tip of my tongue, but he's got, you could look up his paper. I think it's available online, but he's got some charts there on shedding. And, they drop off with treatment and then they rebound within a week or two.
Yeah, I'll definitely take a look into that. I think last two questions, you mentioned that sometimes Trichomonas might present something similar. What is the treatment like for that organism? / what is that organism, I guess?
Okay, well, we'll start with that. So it's a parasite, and it's a single cell parasite. And they're members of that there's other organisms that are similar, Giardia and Hexamata, but they're single cell parasites that are motile, and they live in a very discrete portion of the digestive tract. And in fact, in finches, the only place we can find them is the esophagus, just as it exits from the crop. And so that's their spot that they like. And they cause inflammation, severe inflammation of the crop. And they actually can, in some instances, in pigeons in particular, they can spread to internal organs as well. But they can, like, again, if I was going to try to make a diagnosis of that in a budgie or in a finch, what I would do is take an IV tube and cut it into a very small, and an IV tube is very soft plastic, and I would cut it into a small bit and put it on the end of a small syringe. Take and pull up some saline or even just some water, but probably for Trichomonas saline. I'd drop a little bit, put that tube down into the crop, flush a little saline in, flush a little bit out, put just a drop of that on the microscope slide, put a cover slip on that, and then look same way I look for Macrorhabdus. And those organisms are very active and they're swimming all over the place. And you go, cha-ching, that's what I have here. And in pigeons, they're a normal part of the gut flora in low numbers, but if you see huge numbers, then there's a problem. But in budgies and cockatiels, I don't expect to see them. And so if I see them, then I would use a drug called metronidazole. And I would give that orally and expect with a few treatments that we will have solved the problem.
Yeah, and I think you mentioned before that it's easier to treat than the Macrorhabdus.
Right, yeah. So with pigeons, they use a drug called ranitidazole, and they give one tablet into the mouth, and then it's done.
That is a lot easier.
Yes.
Do you know how
you need
they
to make sure that they're not reinfected? So clean thoroughly, make sure the water supply is not contaminated, the food supply is not contaminated.
and is it?
As they said, they shed cysts that are environmentally resistant.
Is there any disinfectant that they're sensitive to in cyst form?
Well, probably, but I think just elbow grease, just getting rid of the fecal material. And then if you wanted to use bleach or Lysol or something like that, that would, I'm sure, kill them.
Yeah. And I'm assuming fecal oral, they were exposed to another bird who was actively shedding the cysts.
Yes, that's right. Yeah.
Yeah. let's see, vitamin C we already touched on, and I'll get the info from you later on that. That's very helpful because kind of on the same lane of things, do you think that it's worth it to supplement any other vitamins, maybe not specifically to target the Macrorhabdus, but because the bird is ill and needs some kind of supportive care? Um,
Well, it's very common for us to use vitamins in any animal that's ill.
mhm,
Multivitamins in the drinking water
mhm,
are fine. But again, we don't just want to feed budgerigars seeds. We'd
mhm.
like to get them, if they're on a seed diet, we want them to have greens or other things like that. So we can stimulate them to lay, right? But
Yeah, so
they get vitamin A and they get vitamin B and things from greens and other things. And it's an enrichment thing, which may not be the best and when it comes to trying to keep them from laying, but they do enjoy munching on greens. And I know that one thing that budgerigar breeders were quite fond of giving budgies was dandelions. They would give them dandelion greens and also the flowers. And
yeah.
budgies really like those.
Yeah,
Yeah.
I think for most of us now, yeah, all seed diet is well, still common numerically, but by the time people come to me, one of the most important things I do is help people get them onto a diet that actually checks all the nutritional boxes. Otherwise, if it's just seed, it's so deficient in so many categories. But yeah, usually we offer some amount of fresh produce every day, chopped slash with some chunky items, and then we can of course hang some whole leaves. And of course they love and they love their leaves. So
Sure. Yeah.
um, let's
So
see.
if it stimulate, if again, I would think that greens as a pair compared to seeds would be a lot easier to digest. They're not going to be grinding them in their gizzard so much, and that's already going to be painful. So,
Mhm.
um, and if you want, I always tell people, that ask me if they should be giving vitamins or not, and I said, well, you only want to give vitamins if you don't think the diet you're feeding is balanced diet,
Yeah.
and that's a better way to do it. But if you want to give supplemental vitamins, maybe put it in the water once a week or something like that.
Yeah. That's the interesting thing for most of us. They're already on a balanced formulated diet paired with whole foods. So they have a freely available base that's formulated, and then the top they also get fresh produce and different kinds of things. We have noticed that the softness of the food, Nathan and I were talking about this before, that we've noticed that the bird was uncomfortable eating harder foods. So we offered something softer, a little bit of like cooked grain or quinoa and things like that. At least it's the protein is better quality, and that did seem to help. But definitely we'll keep that in mind. Because we've observed that too. Yeah, the hardness of the food is the birds start to struggle if they're really ill. Um, and I think the last question I had was from Jenna that she submitted before she had to go. But what about protozoan disease? This is a little bit kind of separate in topic, but kind of along the parasite organisms that we don't want in our bird.
Right.
Um,
So trichomonas are protozoans
mhm,
and Giardia are protozoans.
mhm,
And trichomonas live in the upper digestive tract predominantly, whereas Giardia lives in the duodenum.
mm,
So a veterinarian might do a crop aspirate,
mhm,
but if they've got fresh droppings, what we say is their fresh droppings are still steaming hot, that we will, with warm saline, put it and look under the microscope, and we might see Giardia running around
mhm,
in there and see that. So as part of a normal well bird exam, a new
mhm.
bird that's coming into an aviary, we'd want to do a crop wash and a fresh dropping wet prep.
Yeah,
And if they've got Giardia or tetrameres, or not tetrameres, Giardia or hexamida or trichomonas, so we would pick those up on either of those with either of those two tests.
yeah, and both are, I guess, antiprotozoan drugs, metronidazole.
Metronidazole. Historically, we used to use a product called dimetronidazole, which is was being used inappropriately, and so it's been taken off the market. But then there's another product called ranitidazole that people use. But metronidazole, it's very bitter, but it's a very effective drug, at least a bit in my experience.
Yeah, I'll definitely pass that along to Jenna. She was the one who asked me, Amy, she only had the budgie for I think a week and a half or something. She said, Amy, the budgie looks sick. And she showed me a photo and I was like, yeah, this bird is definitely ill, not a little ill, very ill. And went to the vet, yeah, dehydrated. Fecal cytology showed large amounts of Macrorhabdus shedding. So amphotericin B treatment 2 weeks, but then a crop wash, they said that there's a lot of protozoa in the crop. So I didn't ask what kind, but
that would have been Trichomonas most likely. So that's just another example where we had both organisms, which both organisms are quite common. So it's wouldn't be uncommon to have both of them present at the same time.
I think the vet said that she was hoping to maybe just not treat for now and but to use filtered water instead of tap water. So I kind of wanted to ask, is that a risk? If we're feeding, if there protozoa in tap water?
Not so much.
Yeah. Yeah. So maybe it's just a treatment. The bird just needs treatment.
At least in my experience, I haven't noticed lots of reinfection after treatment or anything like that.
Yeah. Cool. So that's all the questions I have written down so far. Did I miss anybody? Anyone else have any questions they would like to ask? All right, covered everyone.
All right. Well, everybody asked some really insightful and good questions. So that was enjoyable to have that interaction, and it allowed me to bring out some other points that I had not covered in my talk.
I'm glad. Nathan says, thank you for being here. Yeah, this is really, really exciting, especially because we've talked about your research before in our live Q&As. So it's really exciting to get to hear from you directly and get this much information, all in one place. So super exciting, really informative, really educational. And I really want to thank you for being here because it means a lot to us, even though our community is new and it's small, but I think we're going to do hopefully big things and maybe one of us can be that breeder
all
one
your
day.
enthusiasm and your great clients, I'm sure that this will turn into a wonderful thing.
Yeah, hopefully, one of us can be that breeder one day. We'll breed for proper budgies, good genetics, and strong disease resistance. That's going to be the goal.
Oh, that'll be wonderful.
Yeah. All right.
Well, maybe I'll do that when I get into the aged care facility.
But you're I think you're in the Northeast, right? So maybe I can start a little something and you can visit and then.
Yeah, that'd be cool.
Awesome. Okay, well, I guess this concludes our first official guest lecture session. It's been amazing. Thank you, Dr. Phalen, for coming. Recording will be posted as soon as it's done processing afterwards. And then the recording will go up inside the community first. And then I'll go through and kind of edit out the breaks and things like that, where if we all went to drink water. So I'll edit those pauses out and then I'll post it in the community. And then, if you have any questions, feel free to comment under the post when it goes up. And
well, let me just double check my slides, make sure there wasn't any errors in there before you post it to the world, but to the community is fine. So I'll double check it tomorrow or the next day. So
yeah, no worries. If it doesn't go out to the public, it's totally fine. We can keep it to ourselves and I'll write articles.
I want you to get as much publicity and get as widespread as possible. So I'm more than happy for it to go on YouTube and everything else. Okay.
Thank you so much. Thank you for being here. Thank all of you for being here. And bye.
All right. Well, have a great weekend. Thank you. Bye.
Thank you. Bye.